Kia ora koutou. It's my great pleasure to welcome you here. I'm John Hosking, Dean of Science here at Waipapa Taumata Rao, University of Auckland. It's my very great pleasure not only to welcome you to the university but to the inaugural lecture of Professor Lynette Tippett. What goes on inside our brains is a question which we humans have puzzled over since time immemorial. Today that question is particularly pertinent as the increasing prevalence of many neurological conditions ranging from traumatic brain injury to neurodegenerative diseases is accompanied by growing concern about the impacts on individuals and their whānau. Lynette is at the forefront of multidisciplinary research attempting to bridge between biology and cognition, particularly to provide crucial insights into what goes wrong when neurodegenerative diseases such as Huntington's and Alzheimer's occur. She leads a network of dementia prevention research clinics that illustrates both that multidisciplinary research and additionally strives to combine scientific rigour with manaakitanga. Lynette was born and raised in Taranaki. Seems like a good breeding ground. She studied zoology and psychology here at the University of Auckland, developing a fascination with the brain and what goes wrong with it. This led to an MSc and PhD in psychology and a diploma in clinical psychology, followed by postdocs at Carnegie Mellon and the University of Pennsylvania. Before she was lured back to Auckland. Lynette has taught neuropsychology for more than 30 years and has supervised more than...
Just 30.
I'm taking it from our own bio. And she's supervised more than 80 graduate students. Identifying the complex interactions between brain dysfunction and changes in memory and other cognitive abilities remains at the heart of her work. Lynette is Associate Director of the Centre for Brain Research, and it's great to see so many CBR people here tonight, and was awarded an ONZM in 2023 for her contributions to neuropsychology and dementia. Tonight, Lynette's going to talk about her research in dementia and the brain. Lynette.
Tēnā koutou katoa. Thank you everyone for coming out this evening to come and hear me talk. I've done a lot of thinking about the talk, but I'm still not sure I've got it quite right, but I'll do my best. I began my life, as John said, in Taranaki, in Hawara to be precise, a small town in South Taranaki, and Taranaki definitely punches above its weight for most things. In our small town, I was the youngest in a family of three. Because I was little, I was shorter than my sister at that point, but that changed as we grew up. It was a pretty happy childhood with all the freedoms of a small town where you could basically do anything as a kid. We had a lot of aunties and uncles and cousins about, and family was always pretty special for us. I think we were a working class family. Our dad owned a small car painting business that he worked in, but it just seemed to be the way it was then. It was a happy upbringing. Like most people in our community, I went to the local high school, Hawara High School, which is no more actually, but at the time it had 1,100 students, and most people went there. I was lucky that my sister Gail had gone to university before me. She was the first member of our family to attend university, but it kind of smoothed the way for me. I didn't have to do any arguing. And I just want to acknowledge that my family has remained a very special part of my life. But at the end of school, I came out to Auckland to study, as John said, biology and psychology. And at the end of that undergraduate degree, I didn't really know what I wanted to do. I enjoyed both of them and I didn't know what direction to go in. So I took a very sensible path and left it all behind and went overseas and travelled and had a lot of adventures and made friends for life. And in amongst that time, I did some thinking, and what came to me was that what had really stayed with me from my undergraduate years was what I'd learnt about the brain, and in particular about what happened to people when they had disorders of the brain. So it was like, okay, that's it. That's what I want. That's the path I want to pursue. It was kind of the perfect union for me of biology and psychology, kind of putting together the complex biology of the brain with the complex functioning of people, and with people right in the centre of that. And so it was a pathway that I haven't regretted. So I came back to Auckland University, did my master's, my PhD, and a training in clinical psychology, diploma in clinical psychology, which I think has also impacted the kind of pathway that my research career has taken. And after that, I went to the States and did my postdocs at Carnegie Mellon and the University of Pennsylvania. And that was a really great time. Once I got over a little problem that I had when I first arrived, which was that nobody could understand my accent. So I would go to a restaurant or a bar, and the drinks person would come up and ask what I wanted, and I might say, because it was very hot, I'd like some water, please. And there was a very high chance they would bring me back a glass of Sambuca. When I would introduce myself as Lynette, most people said, "Oh, Denise, nice to meet you." But once I figured out that if I wanted water, I had to ask for water, and I had to reply to the name Denise. After that, it all went swimmingly. No, it was a really enjoyable and extending time in my life. But I was fortunate then to be able to apply for a post that became available back here in the school, or what was in the Department of Psychology, and I returned to New Zealand. And that began my career. And what did happen to be almost 30 years ago. Almost. Now I'm going to do what I was instructed to definitely not do, and that is to acknowledge my partner Michael and my daughter Georgia for their support and encouragement over the years, and for keeping me grounded and making me laugh when things get a bit tough. So some of you will know Georgia from the earliest Christmas parties when she was just a baby in the department, but she's quite a lot older now. Now, thank you. That was so I didn't talk for too long. Okay, now I also, it wouldn't be right not to acknowledge the kind of academic influences I've had from my mentors and supervisors. And the first person who really had a big impact on me was Dr. Dorothy Gromwell, who lectured me at undergraduate and later at master's level. And Dorothy spoke to us about her work with mild traumatic brain injury and the effects of a concussion, and how providing people with appropriate rehabilitation can really make a difference to their outcome. And I was really struck by that work. At the time, she and her collaborator, neurosurgeon Mr. Philip Wrightson, they faced a huge amount of scepticism, because people didn't really believe concussion was anything more than a psychological phenomenon. But it's now accepted that following a mild TBI, it is common for people to have concussion symptoms, and they will usually, with appropriate management, recover. So Dorothy really pointed me in the direction of this work. During my PhD, I was co-supervised by Associate Professor Jenny Ogden and Professor Mike Koublis. And for those of you who know Jenny, she's a very passionate, noisy clinical neuropsychologist, and she really kind of encouraged the development of that side of me, both professionally and in the research world. And she was very talented at working with people with brain disorders. And Mike, most of us will know Mike. He was a brilliant cognitive neuroscientist who studied many things, including visual imagery and laterality and language, and he of course had the gift of language himself, with being able to explain the most complicated concepts so easily, and his writing was equally clear, and I hope I learnt a little bit from him in that time. And finally, Professor Martha Farrer was my mentor on my postdocs in the United States, and Martha is also a brilliant cognitive neuropsychologist, quite quirky actually, but she really challenged me to think differently about how the brain worked and how cognition worked. And in that time, especially at Carnegie Mellon, she actually got me, and for those of you who know me now, this will seem a little unusual, she actually had me doing computational modelling of how the brain works. So technically, I haven't kept up with that, but it was a really interesting time, and it really shifted the way I thought about cognition and how cognitive functions were organised in the brain, and that stayed with me through my teaching of clinical neuropsychology and my research. Okay, so now I'd like to kind of spend the evening taking you on a bit of a journey through my research career. It's a bit of a jerky journey, not a smooth line, and there's not going to be as many stops as I would have liked because I was told I was only allowed an hour and not 2. So there's a few jumps here. But the journey really is about research focused on that interaction between the brain and how people function, their cognition, their emotional functioning, how they function in social interactions and their family, and how they function spiritually as well. And my work has benefited enormously from being multidisciplinary, from my collaborations with other colleagues in psychology and also the Centre for Brain Research, because that's what enables converging methods to be applied to a question, and there's just so much more that can be learnt when you pull those different levels together. Okay, so back in New Zealand for the first time, I was sitting in my empty office, and yes, I once had an empty office. And I'd not long been back, and I had a phone call from this person called Richard Fall, who I didn't know. And he said, "Oh, hello, good, I've been waiting for you to arrive. We need your help." I was like, okay. And that was actually the beginning of a long association of working in the area of Huntington's disease. And there have been many people I have collaborated and worked with over the years in this field. But at that, and I do want to acknowledge distinguished Professor Sir Richard Fall, because he's had a pretty profound impact on various directions that my career have taken, and also because he epitomises that passion for the brain at the same time as keeping people and the person right in the centre of the picture, and our responsibilities to the people that we're working with brain diseases. So thank you for that, Richard. At the time that he called me, he and his colleague Henry Wardvogel were working with brains that had been donated to the Neurological Foundation Human Brain Bank of people who died with Huntington's disease. So they were looking at the brains of these people who'd died, and what they discovered was that there were changes in neurotransmitter receptor areas, or neurochemical, we'll call them neurochemical compartments of a structure in the brain that was known to be important for Huntington's disease, the striatum, but the changes were patchy. Some people lost one neurochemical compartment, other people lost a different one, some people lost both, but it wasn't just to do with the severity of the disease. And they asked the question, and what they wanted me to help with, was to look at whether this patchy finding had anything to do with the symptomatology that people experienced during the course of the disease. And this was a pretty far-out question, actually, because Huntington's disease is caused by an autosomal dominant gene. So you need one copy of this gene, and if you have it, you will almost certainly, with a few exceptions, develop clinical symptoms of the disease. So it's one gene inherited from a parent. In terms of what happens clinically to people with Huntington's disease, there's a triad of symptoms that are associated with this condition. It's most characterised by the motor disorder that develops as people develop symptoms, particularly an involuntary motor disorder that involves kind of career, irregular, unpredictable movements. But also there are problems with cognition and thinking that develop to different extents in different people. And for some people, there is also a mood or psychiatric component that is not just a reaction to having the disease. It seems to be part of the disease process for some people. So that was the triad. And if you looked at the literature then, what you would read is that it's a pretty standard development of symptoms that people experience. But people on the ground working with people with Huntington's disease, they knew all along that actually there's a lot of variability between different individuals, and some of that is accounted for by characteristics of the gene, but some of it is not so clearly associated with the gene. So you can have, for example, symptoms, you can have siblings in the same family who inherit the gene from the same parent, grow up and present very differently at different ages, with different symptoms predominating during the disease course. And we have written several times about a pair of brothers who really epitomised this, that Virginia Hogg and I worked with while they were alive. And they had the same identical characteristics of the gene. They had a difference in symptom onset of 12 years. One of them, who developed symptoms younger, had really marked new development of mood and psychiatric symptoms, a lot of cognitive problems, and later motor symptoms. The other brother, he developed it when he was 12 years older than that brother. It was a very mild course. There was no mood. There was nothing to explain the difference in these two men. Their environments were almost identical. They grew up on the farm, they married like a woman, they both farm. There was so much they had in common. And even in monozygotic twin pairs, you can see variable clinical symptoms. So we have here this clinical variability, and Richard and Henry seeing that difference in the brain. And the question was, did they, could they explain each other, or were they related? Now, this was quite challenging for a psychologist, actually, because I'm trained to interview and assess people while they're alive. And we were being asked to generate these symptom profiles from people who had already died from Huntington's disease. So I didn't want to say to Richard that this was a problem, so we just set about doing this. So we developed a methodology where we could reliably extract clinical history of each individual. We tested this on people who were alive, the methodology, and we were getting reliable results. And we were able to estimate the age of onset, but importantly, the patterns of clinical change that each individual experienced while they were alive, and particularly the nature and severity of motor and mood symptoms. We've never been able to retrospectively get reliable data on cognition. So people are not very good at being able to report cognition, basically. But the motor and mood components were quite clear and reliable. And these are the sorts of things that Richard and his group would generate. You don't have to understand these, but they're slices through the striatum, and they're stained in a way that shows these two compartments. And so there's a compartment called the matrix in this area, and it's sort of a large compartment. And in this one up here on the top right, a lot of that is gone, and what you're left with are these dark patches called the striosomes. In this case, those striosomes have all gone, and you've got the white patches, but the matrix is in good shape, and some people lose both. And we worked in a double-blind fashion, of course, and we'd made predictions about which compartment would be associated with which symptom. And what we found was that the striosome compartment that had connections with regions of frontal cortex and limbic areas, the people who had lost or had damage or poor striosomes were the people who had severe mood problems, both at onset of the disease and at the end stages of the disease. And so what we were able to show was a phenomenal, it was the first demonstration actually of the functional, in humans, of the functional properties of this little neurochemical subsystem in the brain. We've continued that work and been able to also show that Huntington's disease also affects the cortex, and that there's a lot of variation in terms of where in the cortex cells are lost, and that this again relates to the clinical variation between people. So that's been a really rewarding collaboration. There's a lot else went on. I'd love to have talked about my work on TBI actually, but my partner told me last night, no, you've got to chop something. So I did. And so I'm going to leap to this chapter, which actually began about 10 years ago. And it was, it began in the Corrie Lounge at Auckland Domestic Airport. There were some people here who were also present that day. We were on a trip down to Dunedin, and it was about the application for a Centre for Research Excellence, which was to become BRNZ. But I was kind of thinking, why am I on this trip? I wasn't really part of that development. And so I was kind of thinking, this is a bit odd, oh well. And then while we were in this Corrie Lounge, Richard turned to me, and Alan Barber, who's here right now, had a bit of a smirk on his face actually at the time. And Richard turned to me and said, Lynette, how would you like to set up a dementia research clinic? Well, what do you say? Okay. So that was 10 years ago, and this is now. So we began with the help of some funding from the Alzheimer's New Zealand Charitable Trust and the Angus Trust. A small group of us began developing a prototype for a dementia research clinic, and the people who were really central to this are Gary Chung, who's here, who's an older age psychiatrist, neurologist Dr. Kerry Brickell, neuropsychologist Dr. Christina Ilsa, and geriatrician Dr. Phil Wood, and Karen Smith was our research nurse, and Erin Corsten, our human blood researcher. And we spent a long time looking at international protocols, looking at what we would need to do to have a clinic that would be capable of answering questions. And we were supported by Brain Research New Zealand and with support from Peter Thorne and Cliff Abraham to develop these clinics in Christchurch and Dunedin as well. And right now, because BRNZ was not refunded, we are supported by the New Zealand Dementia Prevention Trust, New Zealand Dementia Prevention Trust, and the Hugh Green Foundation, and we also had some funding from the Aotea Brain Project. And that photo was taken at a clinic meeting of the 3 clinics down in Christchurch last year, and there are some amazing clinicians and some amazing researchers who are still keeping this work going. So what's it all about? Well, we have focused primarily on Alzheimer's disease and Alzheimer's dementia, and we are definitely interested in looking at factors that might prevent or slow down the development of dementia. So with normal ageing, you get a bit of a drop-off in cognition, but not too much. People function well, even in their later years. But for a percentage of people who are ageing, their cognition deteriorates, and as well as age, for many of these people, there will be an underlying accumulation of brain pathology. And when that drop-off in cognition reaches this point here, this is the point at which you can no longer compensate for those changes in your memory and your language and your visual spatial processing, and it starts to impact your functioning in daily life. And at that point, it's called dementia. Now we're all hearing we've got a bit of a tsunami coming in the dementia area, and that's not because dementia is, kind of more common and more people are catching it, or whatever mechanism by which people get dementia. It's because there are more and more people living into the older age bracket, and older age is the biggest risk for dementia. But what we know is that along this sort of trajectory here, we have people who are reporting problems, usually with their memory, and we can test and see that those problems are there. They're not performing as well as they should for their age and education-matched peers. And this is a condition, if you like, that's called mild cognitive impairment. So it means there is impairment on testing, but in everyday life they're functioning fine. They can compensate for those memory difficulties, and they're managing to maintain normal functioning. Now, if we were able to, and I should say that not everybody who's on that line, not everybody with mild cognitive impairment progresses to dementia. Some people kind of stay stable, and some people actually improve and kind of become cognitively like their peers again. So it's not inevitable that will progress to dementia. But if we could first identify those people who are going to progress, and second, do something that delays or prevents that progression, we would make a big impact. So even if we delayed the development of dementia by 2 years, by 2050 we will have a 20% reduction in dementia prevalence. If we were able to delay by 5 years, by 2050 there will be a 50% reduction. So it will make a difference to the challenge for health systems and whānau if we're able to slow it down. So our clinics, we were the dementia research clinic. We were told we had to put prevention in there or we wouldn't get anyone coming along. So we added that into the name. And we really are looking to identify factors that affect the likelihood of progression from memory problems to Alzheimer's disease and dementia. And we're doing this in a large cohort of New Zealanders. And we want this cohort to be reflective of the New Zealand community and the New Zealand makeup of our community. And we're particularly targeting people with mild cognitive impairment, but also older adults with normal cognition. We've also got people there who we call subjective cognitive decline, so they are worried, they think they notice problems, but we can't see it on testing, and we include people with dementia. And we're trying to identify a pattern or biomarker signature that predicts progression. So we're doing this in a very rich way. It could be even richer probably, but we're looking at a whole range of functions. We also really want to understand the experiences of individuals and their whānau who are living with mild cognitive impairment and dementia. And we have a lot of work going on trying to identify mechanisms underlying the development of these conditions. So you will hear really that we know all that we need to know about Alzheimer's disease, but it's not actually true. There are some pathways that are direct into the development of Alzheimer's disease, but there are many mechanisms that might lead to the trajectory of developing the pathology, and it's those mechanisms and opportunities to intervene that we're interested in finding. And we are hoping for our participants who in the clinics to be able to be in a position to test a range of interventions when they become available. So at the moment we have assessed 491 participants. We've had 410 people who've enrolled in the clinics and done a full baseline assessment, and they range, as I said, from healthy or cognitively unimpaired controls and through the trajectory of mild cognitive impairment to early dementia. The 3 clinics, as I said, are in Auckland, Christchurch and Dunedin. So we've got almost 300 who have enrolled in Auckland, and the 2 South Island clinics are smaller but but they bring more variation, more variety, and more involvement for the whole country. So far, we've had 255, I think, people complete their second year follow-up, and we've got close to 150 at the 4-year follow-up, and about 60 who've done a 6-year follow-up, and we've got a handful of people who've been with us for 8 years. And we have about an 84% retention rate of participants, and that's pretty good because these people are older, they're 55 years plus, but there's quite a few who are older, and they develop a lot of other health conditions, or their families develop health conditions, and because what we're asking them to do is quite a lot, we're very pleased that we have got the commitment from such a large number of people. So when each participant comes to our clinic, and they come every 2 years on average, they go through a really thorough assessment. They have clinical, medical, lifestyle, neuropsychological, some mood assessments. They undergo a very detailed MRI scan that includes research and clinical scans, and most of them have an amyloid PET scan, which enables us to see whether they have any of the amyloid plaques that are one of the characteristics of Alzheimer's disease. And we also are fortunate to be take blood from our participants. They generously allow us to take quite a lot of blood, and our blood team collect the blood in 5 different tubes, and from that they derive a lot of different blood products, which are then stored in very small ampules and in the cryostore at minus 190 degrees. So that's storage and handling of our bloods, which will enable the testing for blood biomarkers for years to come. Now, there are 2 things, and they do this every 2 years. So there are 2 things that leap out to me from this. One is that we have a huge amount of data, like a huge amount of very rich data, most of which is now captured in a REDCap database. Our scans are elsewhere, obviously, and our bloods are elsewhere, but there are links to those in this database, and that's managed by Leon Greiner very effectively. But the other thing that strikes me, and might strike you as well, is that the participants in our clinic are doing a huge amount for us. It's, 3 visits, sometimes 4, to complete each assessment. The assessments are delivered with skill and kindness, but they are quite demanding on our people. They're older, and as we do have people progressing to dementia, it is a lot that we are asking for and getting from people. And that's why I wanted to talk a little bit about combining science with manaakitanga, because the care of or the responsibility we have for our participants is enormous. We've always placed a high value on excellent clinical assessment, and we have the highest calibre clinicians working in our clinic. We give very detailed feedback to our participants. We provide them with lots of information. We have annual events. So we've always felt like we have looked after our participants. But at this point, I want to acknowledge Dr. Waiora Port. Waiora is an esteemed kuia and doctor, and Waiora has very gently and very patiently walked beside me for many years in my research, both in Huntington's disease and with these clinics. And she's just quietly, in her quiet way, been kind of encouraging me and guiding me and sort of trying to support me in realising maybe and in the need about bringing manaakitanga as a researcher to Māori and to non-Māori members of our research community. And she additionally walks beside all of our Māori participants who come to the clinic. So I want to acknowledge the guidance and the kind of years of unpaid input that Waiora has given. And I also want to acknowledge Dr. Macarena Dudley, who many of you will know has been leading a group of Māori researchers in the development of a theory of dementia, mati wari from within the Māori world, and also in developing a assessment tool for dementia that is valid and appropriate for Māori. Now, I'm not Māori, so I wasn't part of the research group, but I was lucky to be a part of the advisory group. And what that meant for me, actually what it meant was I often had to shut up. I often had to sit in the room and shut up because I wasn't the research leader. But it gave me like an incredible opportunity to listen and experience and absorb how these researchers were working with kaumātua all over the country and how they were showing care and generosity and reciprocity with their participants. It showed me that there's more than one model of dementia and that for Māori it's Wairua that actually changes first. And it also showed me in terms of the cognitive tool, because I worked closely with Macarena on developing the cognitive assessment tool for this. And it showed me in the kind of iterative feedback we had from our participants just how important it was when you are assessing someone's cognition that they can see their world in those materials. Now, I thought I knew that. I actually thought I knew that. But there was something about seeing and hearing the words of those people that just really hit home. And it also has made me reflect on our clinic. And as our participants have stayed with us for a long time, we've all become a little bit aware of the need for us to do more for them. We have to maintain the integrity of the science, but there is many opportunities to do more for them. And our clinical team, I do want to take my hats off to them for their endeavours in this regard. We have students who, some of whom are working on clinic projects, some of whom are not, who really work with people when they come in for an MRI. People come in, they're pretty anxious actually. It's quite a big deal having an MRI and going into the machine for 45 minutes and having that coil, and they do a great job with those. So, you know, our attempts for taking responsibility and reciprocity, we are attempting to improve in that way. And I'm going to show something which puts me at risk of never being spoken to again, because I want to acknowledge our clinic manager and research nurse Jane Govender, who leads this drive towards caring for and being responsible for our participants. She's a role model for everyone, and she brings her own cultural values that align actually with Māori cultural values to the running of the clinic. So I think it has been a realisation for me, and in the kind of work that we're doing, I think it's really important. But I just want to make it clear that we're not perfect, actually. And last year we set up a participant engagement group, and Gary Chung has been running this as for us, or he's part of our team. And we've had really good buy-in from our participants actually to this, and they are not shy of telling us what we're doing well and what we are not doing well. And some things we thought we were doing really well, they were saying, why aren't you doing this? And we were like, we are. But obviously we weren't in a way that was accessible to them. So it's been it's an interesting journey, and I guess it's part of that listening and taking back and listening and taking back. So so it's a whole nother dimension to the project. But what about some of the research that we are doing? But we've now reached a sort of level of numbers that we can start really looking at some interesting questions. And I'm just going to give you 3 brief examples of projects that are going on before, because I do want to get to the sense of self, because it was in the title. and John Montgomery wants to hear about it. In 2020, there was a paper published on modifiable risk factors for dementia, and there are 12 identified risk factors that account for 40% of the prevalence of dementia. And these are indeed all modifiable, and if we were able to remove them all, we would reduce dementia prevalence by 40%. And this was pretty, outstanding new information, because there had been a feeling that we couldn't do anything. If we couldn't do something biological, we couldn't do anything. Now, the risk factors there that are in red are in red because they all align actually with risks for heart disease. They're all cardiovascular risk factors, but they are also risk factors for the vascular system in the brain, for the blood vessels in the brain, the vessels that deliver blood, which delivers glucose and oxygen to our brain matter. And we know that basically what's good for the heart is good for the brain. But the questions we are looking at is, how exactly does the cerebrovascular risk increase risk for dementia? And in particular, is it related to Alzheimer's disease and Alzheimer's dementia? Because there is a small amount of evidence suggesting that it may exacerbate the cascades of pathology, deposition of amyloid and tau. So Alzheimer's disease pathology is characterised predominantly by these clumps of proteins, amyloid plaques and tau tangles. There is neurodegeneration or loss of neurons, but there is a question about whether vascular factors may be a fourth factor that contribute to the cascade that happens. So we're doing a number of projects in this area, and the MRI work is led by Catherine Morgan, who is one of the leads on our neuroimaging platform. And we're looking at a number of different mechanisms, if you like, by which vascular health might impact dementia and, in particular, the development of Alzheimer's disease. But I'm just going to talk about one of these briefly to give you an example of the work going on. So the second one here is related to disease of the small blood vessels and to the accumulation of something called white matter hyperintensities. So in this scan here on the right, you can see these sort of white, kind of blurry patches that are developing. And these are not holes in the brain. These are kind of areas of lower density in the white matter. They happen increasingly with age, and they can be present in cognitively normal people. So they don't inevitably cause cognitive problems. But what we are interested in is whether these hyperintensities are sort of midway between healthy tissue and tissue death, whether they are in any way promoting the cascade of Alzheimer's disease. I think the vascular, before I forget to say it, the vascular elements of dementia are also important for the New Zealand population because they, Māori and Pacifica people have been shown to develop dementia earlier than Pākehā and Asian communities, and they also are overrepresented in all these cardiovascular risk factors. So if we are able to sort out the mechanisms by which these risks are occurring and work towards treatments for those, we may make a real difference to the New Zealand population. This example I'm going to give you of work going on is the work of our PhD student, Cameron Heyman, and he is looking at whether the volume of white matter hyperintensities is related to clinical and cognitive impairment in our sample. And his preliminary findings show that if you look across the clinical groups that we have from healthy control across to increasingly impaired to early dementia, we have, as expected, a volume in the control group. This is a log scale, but there's a linear, significant linear trend showing increasing levels of white matter hyperintensities in those groups. Now, that doesn't tell us whether that's causing the cognitive impairment or how that's interacting. But the next step of his work is trying to see whether the location of these is associated with specific cognitive problems. So that will tell us if they're making a difference or not in our samples. So we're doing this by using software, or collaborating with Flavio Dell'Acqua from King's College London. He has a imaging software that enables us to determine the load for each white matter track in the brain of white matter hyperintensities. And then we'll be able to examine whether the extent of the load is related to the cognitive problems that are associated with that, cognitive functions that are associated with that particular tract. We are also looking at a range of structural MRI brain measures and cognition, and looking to see whether looking at that integration can help identify those at highest risk in those earlier groups of developing Alzheimer's disease. So I'm just going to show you a little bit of Tess Chaffee's work. Tess has her doctoral oral at 8 a.m. tomorrow morning, so she's not here tonight. This work is co-supervised with Dr. Reese Roberts and Ian Kirk, and Catherine are involved in the structural work as well. But basically, what we're seeing at the early stages is when we look at our people at baseline and then look at the people who have converted to probable Alzheimer's dementia at 2 years later, and we then look at the people who haven't converted, we're able to use techniques that enable us to tell what, in this case, cognitive measures are the ones that are reliably contributing to conversion. So which measures at baseline are the ones that reliably tell us who is going to progress to dementia 2 years later. And what we've found is two memory tests, not surprising in one way. A verbal memory test and a visual memory test are the things that load most highly on predicting, and also a measure of executive function, category switching. Now, what this might enable us to do is to have much more refined and targeted cognitive batteries that will then be, easier for clinicians to administer, because it'll take less time, and that we can have some confidence in the accuracy of them picking up those people at highest risk. When we look at the brain regions showing the greatest difference at baseline for those who converted versus those who remained stable, we find a range of areas that include the regions involved in memory, the hippocampal regions, temporal regions, but also the anterior cingulate region of the brain, which is likely to be particularly involved in the category switching task that we've found cognitively. I've got to hurry. I just want to have one quick shout out to our blood biomarker work. This is work being led by Erin Corston. And what's happened in the course of our clinic is that it has become possible to measure really tiny amounts of protein in the blood that have come from the brain. So this is a new thing, really, the ability to detect brain-derived proteins in the blood. It's not easy, actually. It requires highly specialised equipment. Erin has developed a collaboration with Dr. Heinrich Zetterberg, who is a world leader in investigating proteins that can be measured in the plasma of blood using Samoa technology. And you probably, a number of you will have seen earlier this year, there was like a big noise about the fact that there's a blood test that might revolutionise diagnosis of Alzheimer's disease. And what this related to were findings that are using this technology of detecting amyloid and tau in the blood using Samoa technology. And one particular one, the phosphorylated tau 217, was shown to differentiate AD from other conditions at a very high rate. and it was measurable in mild cognitive impairment as well. But it was a big study, but still we need to know whether this holds in the New Zealand population. We need to know what happens. We need to be able to track people over time to see whether the presence of that protein truly results in progression to Alzheimer's disease. And Erin has started the work on looking at the relationship between this tau protein in our participants with their PET amyloid scans. And in this preliminary data, we've been able to show that the people who were amyloid positive for PET were also showing the highest volumes or amounts of the p-tau217, whereas those who were negative were showing very low numbers. But there is this intermediate range that is yet to be sorted out. Now, this is a very small amount of data, and we're continuing with that work. And we will be able to look at our blood samples over time to look at the trajectory of these proteins. Now, there's a move internationally to diagnose Alzheimer's disease just by the presence of a protein. In the United States, there's a real push in that direction. But I hope, and I'll be arguing against that becoming practice in New Zealand, because if you tell someone who's cognitively healthy and normal, who decides, I want to find out if I've got that protein, that they've got that protein, you take away all hope from them. And in my humble opinion, Alzheimer's disease and dementia is only present when the condition is actually causing impairment in functioning. And we know there are people with positive amyloid scans who live their lives with never developing cognitive problems. So this is really exciting work, but a work with a high degree of responsibility. And finally, just sense of self and dementia. This is work that I've been conducting with Professor Donna Addis. Donna was one of my master's students some years ago, and is now a professor at Rotman Institute and University of Toronto. And a lot of our students have worked, I don't know what happened to that. That was perfect when I left home. A lot of our students have worked on this project with us. So, sense of self and dementia. One of the biggest fears associated with having dementia is that the person will lose their sense of self, lose their identity. It's a fear that the person has, it's a fear that the family member has. And it's a common portrayal of Alzheimer's disease, that it is a gradual loss of self. And there's some research that shows family members and caregivers will say things like they're not the person they once were, they're a shell of their former self. And that sort of led to this feeling or belief that there is a loss of self in Alzheimer's disease. Now, Alzheimer's disease is a long journey, I'd like to add. People will be diagnosed in mild symptoms. They will live for many years with dementia. And things change, but the fear that this is something like really prevalent and a part of the disease all the way through is very strong. But there is an alternative view that actually the sense of self, the essential sense of self in dementia, remains. There was a wonderful video documentary that a woman in the States put together, and she had videoed her mother on her journey in dementia, and it was put together and it was very watchable. And one of the things that she commented on was this: the revelation to me is that my mother projects a very strong sense of self with no memory. She's absolutely a full person without it. So her mother had lost her knowledge. She didn't know her daughter was her daughter. She was living in care, but her sense was that she projected this very strong sense of self. And she had made this comment of she had her mother saying this, that I did have a warm feeling, and that simply hit me today, just now, and I'm happy it's here, and I'm not sure I remember where everyone lives, but there's something close that's still with me, and I'm grateful for it. So there is a counter view that maybe self persists. Now, what do we mean, actually, by sense of self? That's a big question. Most commonly think when you're talking about sense of self that you're talking about your identity. And your identity is your self-knowledge, your knowledge about, your attributes, your traits, your preferences, your beliefs and values, your social identities, your roles. So for me, I might have, for example, some features like that. But a second part, a second part of your identity will be made up by your narratives about your life, your ability to kind of weave together a story that encapsulates your life and tells you who you are. But it's not just that, because now this is difficult because it's hard to study, but there is also another, that's, if you like, the content of yourself, but there's another sense of self, which is your conscious experience of being you, of the person looking out through your eyes, what it's like to be behind your eyes. Some people call it your essence. William James called it the I self. And so it's a more complex process than a single thing. People talk about self-continuity being the sense that yourself is the same over time. So you are, in your essence, the same person that you were 10, 20, 30 years ago. Now, one of the big questions is how important are our memories to retaining this sense of self-continuity? Like, can you know that you're still the same person if you can't remember memories from your past? Like, if you've lost your memory of episodes that occur in your life. So I know I'm me, definitely, because I can not only remember, I can re-experience the occasion when I stayed with an American family for Thanksgiving, and in a moment that is too detailed to explain, but in a moment of, I don't know what, I managed to turn on the self-clean function of their new oven while all the Thanksgiving dinner was inside. And the temperature goes up to, 500 degrees, 1,000, I don't know, but you can't open that door until it cools down again. And that was this big formal lunch inside this very large oven. And I can still re-experience how I felt. And I can re-experience hearing my friend's sister on the phone going, "You will never guess what happened." And I can re-experience waking up in the morning, because I stayed overnight, just that filtering through the feeling that something wasn't quite right, and then remembering. So I know that's me, because I can feel that memory. I know what that feels like. But do you know it's you if you can't have access to memories, perhaps less vivid than that, of your past? Can you still feel like you're the same person? We have developed a framework based on the existing literature, and that continuity has always been thought to rely on remembering your past events or remembering your narratives of your life. And that has been the view that's been held strongly. But what happens when you ask people with dementia about their self-continuity? What happens when you say, do you believe you're the same person as you were in your early 20s? What do they say? And in an initial study that we did with AMCI and AD dementia, we found that about 50% of the people said yes, roughly the same, if not slightly more than but the same essentially as our control participants. So they say, yes, I do feel like I'm the same person. And if you ask, well, how do you believe you've changed, and how do you believe you've remained the same? And then you look at the kind of responses that they give, what you find that differs between the AD participants and the controls is that their responses all reflect this I self, this core essential sense of just being you behind the eyes. They're aware that they have, their attributes have changed, what they can do has changed, the roles they have, they're aware that they've changed, but they're very clear that that essential essence of their self has not changed. And they're not terribly articulate about it. They'll say, well, yes, I don't know why, but I just feel like I'm the same. They're very sure that they feel the same on the inside. I've looked at this in collaboration with a UK group who had a cohort of nearly 1,500 people with mild to moderate dementia. And when we look at their response to the yes-no question, I feel I'm the same person I've always been, 79% of them say, yes, I do. So despite having dementia, they say, I feel on the inside like I am the same person. And the 21% who didn't actually were people who were struggling with psychological well-being and lower ratings on living well. They didn't differ in their disease severity. It wasn't about characteristics of their disease. But the striking thing to me is that about 80% are very sure that they still feel the same. So this is making us think that actually, like, we might use autobiographical memories to know that we are who we are, but you don't actually need them to know that you are the same person. And it means there must be other mechanisms that are functioning to give you that feedback that you are the same person. And Odyssey Davis, PhD student in the lab, is looking at those other mechanisms right now. But we know that retaining sense of continuity is good for psychological well-being. So I think there's real scope for interventions to assist with that sense of the person remaining themselves. Not too bad, 7:05. Now, I do want to thank and acknowledge my collaborators, my colleagues, my teaching colleagues, Suzanne, Ian, Paul, Reese, my sounding boards in the department, my clinical colleagues, my friends, my heads of schools for sort of enabling, and they've had to enable me actually in the last 10 years to be on this journey, and to all our professional staff for everything they do, which for me is getting me out of sticky batches most of the time. And also my students who, really drive the research that we do. And I'm sorry I couldn't talk about each and every one of you and the work that you do, but they really make our academic careers come to life. So I'm very grateful for them. Thank you.
Makura pea, makura pea, makue e afie, ki te ara atuku, makuko e afie. Yeah, that's you all.
Oh. I'm speechless.
So I just want to say a few words on behalf of all the brain researchers in our Centre for Brain Research across the University of Auckland, and in fact across all of Aotearoa. Because in a humble way in which she collaborates, she pulls teams together in the most incredible way. And you saw that in every aspect of her work. She puts everyone first. And she's the most wonderful person and the only person who could have developed these dementia prevention research clinics. And when I've known you for almost 30 years.
Almost, yeah.
So she taught me and our research team that just looking at the brain was not enough. You had to go to the community, you had to go to the people. And then we could realise why we were seeing what we were seeing. And that set a foundation that, linking with other researchers, Lynette and I have collaborated ever since you've come back, and taken me on journeys, taking all our research in the Centre for Brain Research. She's one of our rocks. And in fact, by showing the importance of collaborative neuroscientists working together with clinicians, with the community, with whānau, with iwi, has influenced our whole research. It has set us up. That was actually the foundation for our Centre for Brain Research. That was its foundation for getting our core, forming the clinics, and she's the most humble, wonderful person I've known. And thank you, Lynette, so much. And we've got a special taonga to give it to you. And Sonny is going to bless it.
Drop it. Drop it. Do you want to put onto it?
You may wish to explain a little bit about why you chose that.
Well, okay. So, yes, so there was a huge variety of different taonga that we looked at, and I had an idea before we went down what we might choose, but of course we ended up getting something quite different. So this particular taonga means, the meaning behind it is the circle or the cycle of life. And I was thinking to myself, well, Lynette, you can think of all the students that have gone through your supervision and graduated, and you've got a whole another bunch coming through. So it's like a never circle. But this does represent the cycle of life, the circle of life, and we thought that was really appropriate for Lynette. That's about it. I'll pass it over for our co-marshal to bless.
Kia hiwara, kia hiwara. Kia hiwara ki tera tuku, kia hiwara ki tēnei tuku. Kia hiwara ki runga tēnei taonga. Tēnei taonga ka inuitia, te taonga me whakamaumahara mō rātou i hui mai tēnei pō, tēnā koe te mārei kura, nau tēnei taonga ma mātou e inui atu ki runga i a koe. And let this taonga be a reminder of those that you have taught up on the board here, that they also are part of this taonga that we give you tonight. It is my pleasure as a kaumata for the CBR to present you with this taonga. I'm so honoured to be part of you. I've known you for not very long, but I enjoy the times that we have shared up in psychology. Nā no reira, tēnei tamātu inei, kia whakamaua, kia huia, tae ki tēnā mai rāra.
Kia ora. Kia ora. Kia ora. Thank you. Thank you. Thank you so much, Ryan.
So we'd like to finish off singing the Centre for Brain Research Waiata for you. And those of you who know it, which I'm sure is 80% of the audience here, please join in. Tēnā koutou e ngā iwi, tēnā koutou katoa, ko mātou ngā kairanga hou, tino mi haro te roro, te huinga, hiringa ro, te huinga, hiringa ro, te oranga mō ngā tangata tātou katoa, te oranga mō ngā tangata tātou katoa.
Kia ora. Kia ora. Thank you. Thank you, Martha.
So you never know what you get when you come on to an inaugural lecture, especially for giving it. So thank you, Lynette. It was a fantastic lecture, a great recognition of your abilities and your impact. So thank you.
Thank you, Darryl.
Go well, Susan.
Thank you.